A very warm welcome to you all. This is an inaugural lecture, as you know, and I know how much we're all looking forward to it. I'm Anne Greenoff, and I'm head of School of Medicine, and it's my privilege to be introducing our speaker for this evening. Professor Albert Ferro studied medicine at King's College London, during which time he was awarded a first-class honours degree in biochemistry. He has had a prestigious clinical and research training, including in Geneva, the Hammersmith Hospital and the Royal Postgraduate Medical School, and as an MRC training fellow at the University of Cambridge, Addenbrooke's Hospital. He did his PhD in clinical pharmacology at the University of Cambridge under Professor Maurice Brown. He has been a consultant paediatrician at Guy's and St Thomas Hospital since 1996 and a senior lecturer in clinical pharmacology at King's since then. He was promoted to reader in 2007 and then to Professor of Cardiovascular Clinical Pharmacology in 2009. He leads on the teaching of therapeutics to medical students and is course organizer for the intercalated BSc in clinical pharmacology and translational medicine, and leads on the MSc in clinical pharmacology. The main focus of his research is on the role of platelet-monocyte interactions in the pathophysiology of atherosclerosis, and the role of the vascular endothelium in regulating both platelet and monocyte function. The work of his group has shed new light on the mechanisms by which platelets, monocytes, and endothelial cells interact, and how these interactions may go wrong to give rise to atherosclerosis, the underlying cause of heart attack and stroke. We're all very much looking forward to hearing from Professor Ferro give his inaugural lecture, which is entitled Atherosclerosis, A Journey from Blood Cells to the Artery Wall and Back Again. Professor Ferro.
Thank you very much, Anne, for that very generous introduction. Good evening, ladies and gentlemen. I was obviously very pleased to be invited to give this inaugural lecture, which is clearly a high point of being promoted to professor. When I set about putting this talk together, I found it a rather daunting task and quite difficult, because by its nature it is quite different to the kinds of talks that I would normally give, let's say at departmental meetings or conferences. Because on the one hand, you've got to present enough stuff in this lecture to keep the specialist, your colleagues, reasonably interested. And yet on the other hand, you've got to make it reasonably accessible to the non-specialist, the general public, and show why your work is actually important. And I think that's not necessarily a bad thing, actually, because all too often we as scientists get a bit bogged down in the detail of what we're doing. And I think it's actually quite nice sometimes to be made to stand back and have to show people why you think your work is important in the grand scheme of things. In addition to which, of course, it's expected that you put a bit of the personal stuff in, and obviously that's important to me. It's very important in the journey that I've had to get here, but it's obviously not the kind of stuff I normally put into a lecture. So I've tried to put it all together into some kind of synthesized whole, and I hope it makes reasonable sense and keeps people interested. So the title of my talk is Atherosclerosis, A Journey from Blood Cells to the Artery Wall and Back Again. But I am, during the course of this lecture, going to talk a little bit about my own journey and how I've got to this position, really. So just to give an overview, I'm going to start off in the first few minutes, first 10, 15 minutes, giving a potted personal history, my journey here, including how and why I got into cardiovascular research. I'll then focus on the main topic of this lecture, which is atherosclerosis, talking about the scope of the problem, how it occurs, the mechanisms, the consequences, and I'll talk about the work that we've been doing in this area. I'm going to talk a lot about this particular molecule, nitric oxide, or NO, because we've done a fair bit of work on this, and it turns out to be very important. It's an endogenous mediator, which is very important in maintaining vascular health and in preventing atherosclerosis, amongst other things. I'm going to talk about the role of the endothelium, because the endothelium is very important as a producer of nitric oxide. And for those people who don't know, the endothelium is essentially the single layer of cells which lines the inside of all blood vessels. We've done some work on that, and I'll talk about that. And I'll then move on to talk about our role looking at blood cells, blood platelets and blood monocytes. The platelets are normally thought of as being involved in thrombosis, clot formation. And monocytes are a type of white cell, inflammatory white cell, which are involved in inflammation. But it turns out that both of these cell types are also very important in the causation of atherosclerosis, and we've done some work on that. So I'm going to talk about all of that, and I'll end my lecture saying a few words about some novel therapeutic targets, how we might develop new ways for treating atherosclerosis. Well, first to start off, a bit about me. So this is me at a rather tender age, and this is where I was born, Dulwich Hospital, which is sadly now closed. But this is to show that I've always been a King's man, actually, because I was born at Dulwich, which was connected to King's College Hospital, which was obviously part of King's College London. So I've started at King's, and I've, well, I won't say ended my life, but I've come back to King's. And I grew up during the 1960s. I was a child of the 60s. And what were they like? Well, unfortunately, I don't really remember, for two reasons. First of all, it was a long time ago, and secondly, I was quite young at the time. So this is me at a slightly less tender age, in front of my dad's car, which was a rather cool Volkswagen Beetle. And I went to primary school here at Oakfield School in Dulwich. And what I do remember from the time is that a lot of people smoked, and including one of my teachers who used to smoke in class. Extraordinary, really, but that's the kind of thing that used to happen then. And I think smoking was very much more of an accepted thing at the time. Smoking was advertised widely. This was a common advert for a particular brand of cigarettes that abounded at the time, with lots of beautiful people in it. And many of our idols, our screen idols on the silver screen and on the small screen, also smoked. So it was a very accepted thing at the time, and that's obviously very relevant to the lecture that I'm going to give. So that's my one abiding memory of my primary school days. And from there, I moved on to secondary school at the City of London School here. And in my initial time there, I was very interested in languages, and I thought I'd probably end up doing something in the language line, being a linguist of some sort. By and by, I got more interested in the sciences, especially the physical sciences, physics, chemistry, and also mathematics as well. I didn't come to biology until quite late, actually. I decided I wanted to do medicine, but in order to do that, I had to do a biology O-level. And there's one person who helped me through getting biology O-level, and he was actually not one of my teachers, but he was one of my classmates. This is us in the third form, so I would have been 12 years old here. You'll notice the cherubic little chap in the middle there behind the teacher, which is yours truly. And next to him there is a boy called Colin. I'm not sure if he's here. He was going to come, so many people I can't see. But hopefully he's made it, and if you're here, Colin, you're the person who helped me through biology O-level, sorry. So very many thanks for doing that and for helping me. He's my oldest friend and was my best friend at school, so I've got a lot to thank him for. And from there, I moved to study medicine. I came back to King's. This is the Strand campus of King's College, which is where preclinical medicine was at the time. Since then, it's moved out. It's mostly based here at Guy's, actually. But at the time, that's where I did anatomy, physiology, biochemistry, pharmacology, including my intercalated BSc in biochemistry. And from there, I moved to my clinical studies at King's College Hospital in Denmark Hill. And during my clinical time, I'd pretty much decided that what I wanted to do was cardiology, or something in the cardiology line. And this was very much down to two rather inspirational teachers who are shown here. Dr. Samuel Oram on the left and Dr. Graham Jackson on the right. Dr. Oram was a very prominent, eminent cardiologist at the time. He'd actually retired by the time I was at King's, but he was still giving lectures to the students, and I found his lectures very inspiring. And Graham was the other person. He was a newly appointed consultant at King's at the time. And again, I found his lectures and his bedside teaching extremely interesting. And the other reason I have to thank Graham is that later on in life, I came across him again when I returned to King's as a senior lecturer. He was a colleague. And it was through him, really, that I met my future wife, Alethea Cooper. Alethea worked for him as a researcher. And Graham was very instrumental in supporting Alethea through her studies. She was studying part-time for degrees in health psychology. She did an MSc followed by a PhD part-time. She did them with Prof. John Weinman, who I think is here in the audience as well. So Graham was very supportive of her as well, so we both have a lot to thank him for. So, by the time I graduated, and that is me there, as you can see, by that time, I developed some facial hair, a pair of glasses, and some rather bad hair on the top of my head, actually. I decided that I wanted to do something in the cardiovascular line. So I did my general training, as was customary at the time, house officer, SHO, registrar, and then decided that it was time to apply for a research post to do a PhD. And I saw an advert in the BMJ to do a PhD with Dr. Peter Weisberg, who at the time was in the clinical pharmacology unit at Addenbrooke's Hospital. And I went to see him. Peter was very encouraging, and he encouraged me to apply. I duly did, and got to the interview. And on the interview panel, as well as Peter, was Professor Maurice Brown, Professor of Clinical Pharmacology at Cambridge, who's here in the audience, and I'm very pleased to see him. I didn't get that job. Somebody else did, which I was very peeved about. But Maurice was very nice to me. He said, you know, I'd be really interested in you doing a PhD with me. And we duly applied for an MRC training fellowship. We got that. And I spent the next 3 years working with Maurice. And this ended up in the production of a thesis called Beta-adrenoceptor cross-regulation in the human cardiovascular system. I'll talk more about beta receptors when I talk about the main focus of this lecture, but that's what the thesis was entitled. And Cambridge was a very useful time because I learned some very good generic skills. I learned how to eat healthily. By the way, you'll see there's one chap who appears in quite a lot of these photos who's also in the audience. He's Pankaj Sharma, Dr. Pankaj Sharma, who's now a consultant neurologist. And the reason he appears in a lot of these photos is not because he was my only friend, but because he was the man with the camera. And I never carried a camera around and took pictures, but Pankaj did. And obviously he wanted to ensure that he was in as many pictures as possible. So this is us learning some good generic skills, how to dress for May balls, how to listen to concerts in the grounds of Caius College. And this is us learning about the academic life. In other words, traveling on conferences and that sort of thing. And you can see that we did slightly stray outside of the conference hall on a couple of occasions. But good generic skills. Having finished my PhD, I then returned to clinical training in cardiology for 2 years as a registrar in Bristol, and then came back to clinical pharmacology in 1995 as a senior registrar at the Hammersmith Hospital, where I worked in the Department of Clinical Pharmacology, which was headed by Professor Donald Davis. And I worked in particular with two eminent clinical pharmacologists, Professors John McDermott and Martin Wilkins, who really inspired me. And I must say, between Maurice and them, you know, I really got into clinical pharmacology, which is certainly not something that I'd thought about as a career option before then. But I really got into it, and I worked for John doing some further research. I was only there for about 10 months, actually, before I then got recruited to come to King's, or UMDS at the time, United Medical and Dental Schools of Guy's and St Thomas's Hospitals. So that's St Thomas's on the top, that's Guy's on the bottom, with a McDonald's at the entrance, which is again very appropriate to this lecture. So Jim Ritter, Professor Jim Ritter, who was the head of clinical pharmacology, recruited me, and I worked in the department from 1996 onwards. And Jim is in the audience. I'm very pleased to see him as well. And he's been very much a mentor to me over these last 16 years. He retired 3 years ago, but nonetheless, he remains a very good friend and sounding board. And all during my time with him, I found him a very, very good and useful and nice colleague. So I have a lot to thank Jim for. Thanks very much. And so I was initially based at St Thomas's for my research. And more latterly, I've been based at this building, which is the Franklin Wilkins building of King's, which is at the Waterloo campus. So that's where our research labs are. That's where my office is. But I continue to do clinical work as a consultant physician at Guy's and St Thomas's Hospital. So that's a little bit about me. Now I want to come to the subject matter of the lecture, which is atherosclerosis. Now what that means is hardening of the arteries, specifically due to the occurrence of atheromatous plaques. And essentially, this is what an atheromatous plaque looks like. This is a cross-section through somebody's coronary artery that's affected with atherosclerosis. And what you can see is the normal channel through which the blood will pass, which is normally this big, is very severely narrowed down to almost a slit by this big lump here, which is called an atheroma. And you can see that the middle of it is full of fat, full of lipid, and overlying it is a fibrous cap. It turns out the fibrous cap is very important in protecting the patient, actually, because if that fibrous cap ruptures or fissures, it exposes the underlying lipid. The lipid is very thrombogenic, it's very prone to form clot on top of it. And that's the kind of situation where the artery will clot off completely and you can get the complications such as heart attack and stroke. So the integrity of the fibrous cap is very important, and it can rupture under conditions where the artery is under particular strain, and especially if there's a lot of inflammation going on in the plaque. And this is just showing the it's a section through somebody's aorta post-mortem that was severely affected by atherosclerosis. And you can see the lipid all the way down the artery, and there's areas of overlying clots as well. So that's what it looks like. And the monocyte is very important in the causation of atherosclerosis. Normally, monocytes just circulate in the blood, and they don't attach themselves to the endothelium. But under conditions where there's endothelial dysfunction, if the endothelium's not working properly, if it's damaged, inflamed, as happens if you're a smoker or have got other cardiovascular risk factors, high cholesterol or whatever, then what happens is the monocyte can then stick to the endothelium, and then it can actually migrate through into the blood vessel wall. Once it migrates through, it transforms into a cell called a macrophage. And the macrophages can take up cholesterol like anything. They take it up in the form of LDL particles, low-density lipoproteins. These are basically particles which are just chock full of cholesterol. They can be taken up by macrophages. The macrophages become swollen with lipid, and they become what are called foam cells. And by and by, you get more and more foam cells accumulating, and that causes the lipid core of the atheroma to grow and grow. You get a lot of other changes occurring as well. So, for example, you've got collagen synthesis, which is important in the fibrous cap formation. You get a lot of inflammation going on. You get oxidative stress with the production of reactive oxygen species. You get proteolysis. You get new blood vessels growing in as well. And all of those things contribute to the growth and the development of the atheroma. Now, I said that smoking was the most important risk factor for atherosclerosis. And I also said that in the 60s, when I was growing up, a lot of people smoked. And that's certainly true. Since the 70s, rates of smoking have declined dramatically, so that by 2010, only about 20% of the adult population were smokers. And that compares with about 60% of men and around 40% of women in the 50s and 60s. So smoking rates have declined dramatically, and in parallel with that, you can see that cardiovascular deaths, both in men here and in women here, have also declined over the same time period, from the mid-70s onwards. But they've not declined as much as you would have predicted from the reduction in smoking. And I think there's a couple of reasons to explain that. First of all, diets aren't what they used to be, and that's evident from the increase in obesity, especially childhood obesity, which is obviously very worrying. So the availability of takeaways and fast food joints and their uptake has certainly proliferated over the last few decades. This is a rather extreme example, which I came across on the internet. It actually, bizarrely, I found it whilst I was looking for somewhere to go for our team dinner. And for some reason, Google returned this, which is called the Heart Attack Grill. I've never heard of it before, but apparently it's achieved quite a bit of notoriety in its local area. It's in Las Vegas, Nevada, and it serves such delicacies as Dr. John's breakfast prescription, which consists of a donut and a packet of cigarettes. You can get burgers in different sizes, single, double, triple, and even quadruple burgers, which go under the names of bypass, single bypass, double bypass, triple bypass. They're served to you by waitresses who are called nurses, And if you manage to chomp your way through a triple or a quadruple bypass burger, you then qualify for a free wheelchair ride with one of these ladies to your car outside. This is obviously rather an extreme example and not one to illustrate a general point, but the general point is that diets are certainly deteriorated in the last few decades. And the other thing, of course, is that we've developed new labor-saving techniques so that we don't really need to move as much as we used to. The remote control is a classic example of that. And so you can do all kinds of things without moving from the comfort of your own armchair, including walking the dog, apparently. So evolution's taken a bit of a funny turn in the last 50 years or so. It's taken 2.5 million years, give or take, for humans to evolve from quadrupedalism to bipedalism. And in the last 50 years, there's been a tendency to evolve from bipedalism to, I don't know what the word is, zeropedalism or whatever, with unhealthy diets and so on. So I think those changes have contributed to some degree, negate the beneficial effects of the reduction in smoking. Having said that, it's not all lifestyle and environment, but there are other things which you can't do very much about. So atherosclerosis does have a genetic basis as well. So this is a big study published 2 years ago in Nature Genetics, which basically showed the presence of a large number of genes which contribute to atherosclerosis. And obviously, if you inherit those genes, then you're more predisposed, and there's not a lot you can do about that. So the scale of the problem remains very large. And just to give you a couple of summary statistics, in 2010, more than 65,000 people died from coronary heart disease in England, and that was more than any other disease. In 2006, coronary heart disease cost the NHS £3.2 billion. And if you take into account the total cost to the UK economy, so that's NHS costs, costs of days off work and other economic costs, the total was something like £9 billion. So it remains a big problem, not just in human terms, but in economic terms as well. So we do need to understand the disease better, and that will help us to develop better treatments. Now, I talked about the endothelium. I mentioned it earlier on, and just to recap what it is, it's a single layer of cells which lines the inside of all blood vessels, and you can see it here. And endothelial cells produce a variety of mediators, some of which are beneficial, some of which are detrimental. And there's a balance there. In a normal, healthy endothelium, the balance is in favor of the beneficial factors. But where the endothelium becomes inflamed or damaged or dysfunctional, then the balance can tip the other way. One of the most important beneficial factors is this molecule nitric oxide, and we've done a fair bit of work on that. And just to tell you a bit about nitric oxide, it's produced in the endothelium by the action of an enzyme called eNOS, or endothelial nitric oxide synthase, which catalyzes the conversion of this amino acid, L-arginine, to L-citrulline. Nitric oxide is produced as a byproduct, and it can diffuse to various tissues to cause its effects, which are largely mediated through the generation of this molecule, cyclic GMP. So in the smooth muscle, that causes relaxation and vasodilatation. In the platelets, that causes inhibition of platelets, which is beneficial in terms of preventing thrombosis. And in the monocytes, it inhibits them from adhering to the endothelium, which, as you'll remember, is a key event in the pathogenesis of atherosclerosis. So when I started at King's, I returned to this subject that I'd studied in my thesis, which is the beta-adrenoceptor. Now, just to explain what an adrenoceptor is, adrenoceptors are receptors for the neurotransmitter noradrenaline and the hormone adrenaline. Noradrenaline is a neurotransmitter produced by the sympathetic nervous system. Adrenaline is a hormone produced by the adrenal medulla, and many people will recognize them as the fight or flight mediators. They act in the tissues by binding to these adrenoceptors, which then signal within the tissues. And there are two types called alpha and beta, and within each subtype, there are subtypes still. On the blood vessels specifically, there are two major forms. There's the alpha-1 and the beta-2 adrenoceptors, which are shown in blue here. Now, the green triangles are supposed to denote noradrenaline or adrenaline. And what happens when noradrenaline binds an alpha-1 adrenoceptor is it causes the blood vessel to constrict. The opposite happens if adrenaline binds a beta-2 adrenoceptor. That causes the blood vessel to dilate. And when I joined the department, a colleague of mine who's the clinical lecturer at the time, Dr. Matthew Dawes, who worked with Jim Ritter and Phil Hovienchek, had showed that at least part of that beta-2 dilatory response was due to the production of nitric oxide. So I decided to look at that in more detail, and I wanted to look in endothelial cells. Does this actually happen when you stimulate with beta-2 agonists? Does that cause nitric oxide production? And what's the mechanism by which that occurs? So we studied that in human umbilical vein endothelial cells. So these come from umbilical cords, which are normally discarded after labor. So they should be in fairly plentiful supply. That's not always been the case, because often the rate at which they're chucked into the bin is greater than the rate at which we manage to get there to grab them first. But luckily, a few years ago, my twins were born. And there they are, Joshua and Louis. And we said, Alethea and I said to the doctors and midwives at the time, could you please make sure we hang on to these umbilical cords, because Albert needs them for his research. Alethea was promptly whisked into theatre for an emergency Caesar, but to her credit, she said to them, please can you make sure they save the umbilical cords? And they did. So always the scientist and dedicated to my research, so I'm very grateful. And we got these, which are their endothelial cells, which not only have gone into the family album, but which have been used for the purpose of our experiments. And essentially, this is the results looking at what happens when you stimulate beta receptors on endothelial cells. This is just measuring nitric oxide synthase activity. And this is just to show that isoprenaline, which is a stimulant of beta receptors, causes an increase in NOS activity. And the same happens with these substances, which are basically downstream activators of the beta-2 receptor. So essentially, we showed that indeed these endothelial cells do produce nitric oxide in response to beta-2 stimulation, and you could abolish that completely if you co-incubated them with this substance called LNMMA, which is an inhibitor, a specific inhibitor of nitric oxide synthase. I'm not going to go through this data because I think, it just to make the point that this is the kind of experiments that we as pharmacologists do. We do dose-response curves. So in other words, we take a bit of tissue, we string it up in an organ bath, we add drugs at different concentrations, we measure the response, and then we add inhibitors of this pathway or that pathway and see what happens to the response. And doing that enables us to dissect out pathways through which things occur. And essentially, this was what we worked out. It took a few years and a few papers along the way, which were well cited. And most of this work was actually done by Lindsey Queen. Lindsey was my first research assistant. She joined me very soon after I came to the department. She came as a pre-doctoral research assistant on an MRC project grant. She stayed with me for almost 10 years, I think it was. 10 years? Yeah, okay. She first as a research assistant, then my PhD student, and then my postdoc for a few more years. So she does hold a special place in my heart, and she's here today, so nice to see you, Lindsey. In summary, we found that the beta-2 receptors work through two pathways, both of which converge to cause phosphorylation of eNOS, the addition of a phosphate group onto this enzyme, eNOS, on a particular amino acid called serine 1177. And that, in turn, that phosphorylation event causes eNOS to become activated, and then that triggers NO biosynthesis. We found an interesting twist in as much as nitric oxide, once it's generated, actually causes a change in voltage in the plasma membrane. It causes it to become hyperpolarized. And that actually causes L-arginine to be taken up more avidly. And you'll remember L-arginine is a precursor for nitric oxide. So actually that drives even further NO biosynthesis. So you get a kind of interesting positive feedback loop that happens once this whole pathway is triggered. But that was published, we got some good papers out of it, and we got some good grant funding to take our work forward. And having worked that out, we became interested in the more broader picture, rather than this one particular pathway. We became more interested in what is it that controls eNOS itself. And there were a few things known about it at the time. We know that eNOS can be stimulated by shear stress in the blood vessel. It can be stimulated by certain chemical mediators, such as beta receptors, beta agonists, as I mentioned. And we also know that there's a whole lot of proteins that associate with eNOS, as shown here. And we did some further experiments which showed two novel associations which weren't recognized before. We managed to show that these two proteins, beta-catenin and beta-actin, also associate with eNOS, and those associations are very important. I'll talk more about those in a minute. The beta-catenin work was largely done by Nan Chen, who was a PhD student who worked with me from Peking University, who's now returned there. The beta-actin work was very largely done by Professor Yongji, Dr. Yongji as he was, who came to work with me under a Wellcome Traveling Fellowship and is now a professor of pharmacology in Nanjing. But essentially what we found is that beta-catenin turns out to be an important mediator of the effects of nitric oxide on gene transcription. What do I mean by that? Well, nitric oxide seems to have both short-term and long-term effects. Some of the short-term effects I've talked about already. It can cause vasodilation, it can inhibit platelets, inhibit monocyte adhesion, and so on. But it also has longer-term effects as well. It causes remodeling of the vascular wall. It inhibits atherosclerosis, at least in animal models. That's been shown again and again. And we think that those longer-term effects, or many of those longer-term effects, are due to the fact that nitric oxide alters the expression of certain genes in the blood vessel. And we showed that beta-catenin seems to mediate at least some of those. The other one, beta-actin, turns out to be a major regulator of eNOS activity. In other words, it has to associate with eNOS before eNOS can become activated. If beta-actin does not associate with eNOS, eNOS can't become activated in response to any of the stimuli that we know about. So this is beta-catenin. So we have some pictures here of endothelial cells once again, and these are resting endothelial cells. And what we've done here is to stain for beta-catenin using a technique called immunofluorescence. And you can see that most of the catenin is actually around the edges of the cell. And that shows you one important function of beta-catenin, which is that it holds cells together, or it contributes to holding cells together. It forms part of what's called the adherens junction. You also see that there's some beta-catenin that stains up in the cytoplasm of these cells, but none in the nuclei. This is what happens if you stimulate these cells with agents that activate the nitric oxide system. We've got different agents, salbutamol, adenosine, histamine, thrombin, all of those activate the nitric oxide system. And you can see that what happens is that beta-catenin migrates into the nucleus. And we did a whole lot of further experiments which showed that that translocation event is associated with changes in gene expression, in gene transcription. And just to sum that up here, nitric oxide, and we're still working out the mechanism by which that happens, nitric oxide somehow causes beta-catenin to translocate from the cytosol to the nucleus, where it binds this family of transcription factors called TCF-LEF. And then TCF-LEF can then, in turn, bind the genome, where it activates gene transcription. Does that give rise to functional consequences? And the answer seems to be yes. We've looked in one particular area, and that is the area of what's called angiogenesis. This is the process by which new blood vessels form from pre-existing blood vessels. And it turns out the endothelial cells are very important in angiogenesis. And you can show this in vitro, actually. If you seed endothelial cells onto a medium called matrigel and give them a few days, what happens is the endothelial cells, by and by, form tubes, analogous to what they would do in vivo when they're forming blood vessels. And they form these networks, which you can see here. So this is using normal endothelial cells, and this is what happens if you knock down beta-catenin. Essentially, you can see the whole network system is different. It seems to be impaired. There's much less of it than is occurring in the wild-type cells. And you can measure this using special software. And this is essentially the results graphed up. So what we have here are wild-type endothelial cells, and you can see that in response to any of these agents which stimulate the nitric oxide system, then the length of these tubes goes up significantly. And that's completely abolished if the beta-catenin is knocked down. Here we've looked at tube length, but exactly the same thing happens if you're looking at number of tubes or number of junctions. And essentially, loss of beta-catenin is giving rise to loss of ability of nitric oxide to stimulate angiogenesis. And we think that's due to the effects on gene transcription. We're looking into this further, and we're also looking at other possible downstream effects of changes in gene transcription. The other protein I mentioned was beta-actin, and we started off looking at this in platelets. Now, beta-actin is a structural protein in many cells, including platelets and endothelial cells. So on the one hand, it maintains the structural integrity of the cells, and in platelets specifically, changes in actin are associated with changes in platelet shape. Now, shape change is actually a very integral part of what happens when platelets become activated. And that's shown on this scanning electron micrograph here. So here you've got platelets, which are normally fairly flat, disc-shaped. They don't have these protrusions, but when they become activated, what happens is they develop these protrusions, they can adhere to the underlying blood vessel, and you can see that here. This is a white cell that's just passing by here. And changes in actin underlie all of these shape changes that occur. But essentially, we showed that there's one particular form of actin, beta-actin, that binds to enos and controls its activity, and that is globular beta-actin. The normal form, which forms the cell architecture, is called filamentous actin, and that's formed of lots of bits of globular beta-actin stuck together. So filamentous is essentially polymeric beta-actin, whereas globular is monomeric. And it's the globular form which can bind enos. And what we worked out after a lot of experiments is that when you stimulate enos with an agonist, what happens is that two proteins associate. There may be others, but these are the two that we've identified, globular actin and this one called heat shock protein 90. That association causes enos to become activated, and when that happens, that in turn triggers HSP90 to become degraded, to become broken down. That then triggers the release of globular actin, and then the whole system resets itself. So we identified that as a major regulatory mechanism for enos. We published this in a good journal, PNAS, Proceedings of the National Academy of Sciences. Well cited again. This was originally done in platelets. We subsequently showed exactly the same thing happens in endothelial cells, and we published that in the Journal of Molecular and Cellular Cardiology. Okay, so I've talked a little bit about endothelial nitric oxide, and a lot of groups, a lot of people have done work on this, and we contributed to the literature. It's formed by enos, regulated by multiple factors, including shear stress, chemical mediators, and a number of regulatory proteins. We also showed that beta-catenin is an important novel mediator of the effects of NO on endothelial gene expression, and we've shown some important effects on angiogenesis. We also identified beta-actin as essential for enos activation, both in endothelial cells and platelets. But having started work on the platelets, we decided that we wanted to look in more detail about nitric oxide, which is produced from platelets, because a lot of groups, as I said, have looked at the endothelium and endothelial-derived NO. But not many people have looked at platelet-derived NO for various reasons. Firstly, the platelet is much smaller than the endothelial cell, and it produces much less NO. And in fact, there's been a lot of controversy over the years as to whether platelet-derived NO is actually of any physiological importance. But having said that, we know that although the amount produced by each platelet is small, platelets are present in huge amounts in circulating blood. So per ml, there's about 100 million platelets or more. So in sum, you'd expect a lot of nitric oxide to be produced. And indeed, what we also expect is that if the platelet is producing NO, it might have some important local effects around the platelet. Especially, it might inhibit platelet aggregation or indeed promote platelets to disaggregate. And it might also inhibit platelets from interacting with other cells. And the particular cell that we're interested in, and I'll talk about later, is the monocyte. We also had the thought that measuring platelet NO is rather simpler than measuring endothelial NO in vivo, because all you have to do is take a sample of blood, isolate your platelets, and you can measure it there in the test tube, whereas it's much more difficult to do that with endothelial cells, which are stuck down to your blood vessels. So it may actually be quite a useful surrogate for measuring what's happening in the endothelium. And we thought about looking at that as well, and that might be quite useful in disease states. But firstly, to come back to the question of, does platelet-derived NO do anything useful? So this is some experiments we did with Professor Clive Page in pharmacology. This work was again done by Lindsey in association with Bill Paul, who was a postdoc of mine at the time. And this is looking at a rat model of thrombosis. And what we do here is inject these rats with radio-labeled platelets, and we can measure the accumulation of these platelets in the thorax, in the thoracic cavity of the rat, in response to a thrombogenic stimulus, such as thrombin. So on the graph here, when the graph goes up, that represents platelet aggregation, and when it goes down, that represents platelet disaggregation. So you can see that in response to thrombin, you get a good aggregatory response, and then the platelets disaggregate. And also shown here is what happens if you co-administer this inhibitor of nitric oxide synthase, LNMMA. And you can see at the higher doses here, you don't really have any effect on platelet aggregation, but what you do have a marked effect on is platelet disaggregation. So this is suggesting that platelet-derived NO is actually having an important effect not on platelet aggregation, but actually on platelet disaggregation. It's promoting platelet disaggregation. So that's all to do with platelet-to-platelet interactions. But I mentioned platelet-monocyte interactions, and we looked at that as well. This is what's called a flow cytometry plot, a dot plot. And essentially, all you need to know from this is whatever's in the top right-hand corner here, it represents monocyte platelet aggregates. And essentially, this is done in the absence or presence of this NOS inhibitor, LNMMA, and you can see that the number of MPAs, monocyte platelet aggregates, goes up dramatically. So this is again showing that platelet-derived NO is very important in inhibiting the formation of monocyte platelet aggregates, and I'll return to that later. We then wanted to look at what happens to platelet NO in various disease states. So again, work from Lindsey in particular, looking at patients with type 2 diabetes. What we've done here is to take blood from diabetic patients and from healthy control subjects, and we've measured platelet NOS activity. And what this is showing is that at baseline, you get a certain level of platelet NOS activity, and that increases in response to standard agonists. If you look at the platelets from diabetic patients, however, basal NOS activity is pretty much gone, and it doesn't go up in the same way in response to these stimuli. So this is showing that in type 2 diabetes, you have an important defect in the ability of platelets to produce nitric oxide. We showed the same thing in patients with established coronary heart disease. This is work from another MD student of mine, Dr. Peter Okhane, and essentially again showing that in normal healthy subjects, you get a good increase in NOS activity from the platelets in response to standard agonists, completely blunted in those patients with coronary heart disease. And now work predominantly from another PhD student of mine, Dr. Eugenia Kalia. This was looking at hypertensive subjects. Similar pattern, basal NOS activity in the platelets tends to be lower in the hypertensives, not significantly, but it tends to be lower. But in response to stimulation with either of these agents, complete blunting in the hypertensives compared to the control subjects. So these are three situations, type 2 diabetes, hypertension, established coronary heart disease, where we know that patients are more prone to develop thrombosis. In the case of hypertension, we know that there's a relationship between the level of your blood pressure and your risk of developing thrombotic events. So on the left here, we've got stroke. On the right here, we've got coronary events. And essentially, the higher your blood pressure is, it's shown for diastolic pressure here, but the same is true of systolic. The higher your blood pressure is, the more at risk you are of developing thrombotic events. And the theory goes, or our theory, is that deficient production by the platelets of nitric oxide may contribute to this. And indeed, there may also be a reduction in the sensitivity of platelets to nitric oxide. So it's not just what they produce, but it's also, even if you add it from outside, they may actually be less sensitive to NO. And we've shown some evidence for that as well. Back to the monocyte platelet aggregates. So this is where platelets stick to monocytes. And this is shown in these fluorescence microscopy pictures here. So what we've done is use immunofluorescent antibodies to label monocytes here in red, platelets here in green, and if you overlay the two, you can very nicely see that the two clump together. So these are monocyte platelet aggregates. And we looked to measure the number of MPAs in the circulation of patients from our clinic. So we run a hypertension clinic, and we recruited over 100 patients, measured their blood pressure, took some blood, and measured their circulating monocyte platelet aggregates, and found there was a very close correlation. Whether you looked at systolic pressure or diastolic pressure, the higher their pressure was, the more MPAs there were in the blood, and that was very highly significant. So MPA levels increase with blood pressure, and that seems to be related to an increase in platelet activation as your blood pressure goes up due to changes in both NO production and NO sensitivity. So we know that hypertensive patients are more prone to atherosclerosis. We know that hypertensive patients also have higher levels of MPA. Is there a causative relationship there? Our theory is that if the monocyte binds to a platelet, then that monocyte transforms into a more pro-atherogenic type, which then predisposes to atherosclerosis. So the question is, does MPA formation promote atherosclerosis by causing monocytes to become more pro-atherogenic? Well, we did some mechanistic experiments to look at this, and what we have here is a layer of endothelial cells in a plate, and we've stained their nuclei blue using this dye here. And we've added monocytes on top of this endothelial cell layer. And we've looked at two different antigens on monocytes, CD14, CD16. Now, all monocytes express CD14. That's present in all monocytes. But CD16 is a marker of monocytes which are more pro-inflammatory. So if they express CD16, they're more likely to give rise to inflammation. And what you can see here is what happens if you add monocytes alone or monocytes and platelets. And one thing you notice is that if you add the platelets as well, you get many more monocytes expressing CD16. So suggesting that monocytes are becoming more pro-inflammatory if they're incubated with platelets. And this is looking to see how many monocytes adhere to the endothelial cell layer. And you'll see that in the presence of the platelets, you get many more monocytes adhering to the underlying endothelium, and that's graphed up here. So this is showing that essentially platelets are causing monocytes to shift their phenotype to become more pro-inflammatory and to adhere to the endothelium. And that's obviously key to the whole process of atherosclerosis. And we wanted to take that further into patients. Could this be a useful thing to look at in patients? So we recruited some more patients from our clinic. And one of the standard things that we do in the clinic to assess somebody's cardiovascular risk is to use these cardiovascular risk charts. There are other forms as well, different sorts, and there's debate as to which one is the best. But this is a widely used one, the JBS2 charts, or Joint British Societies 2. It takes into account whether a patient is diabetic or non-diabetic, their sex, their age, their blood pressure, their lipid profile. So if you took me, for example, well, I'm a non-diabetic man, I'm 51, my systolic pressure is 120 or thereabouts, and my total cholesterol to HDL ratio is just above 3. So I sit about there, which essentially tells me that my 10-year cardiovascular risk is somewhere between 10 and 20%. So we use that in the clinic quite a lot, and we did that on these patients that we recruited. And we also, this was work done by Gabriella Pasquale, but done with a research ultrasonographer, Dr. Benyu Zhang, who works with Phil Hovyanchek. We also imaged their carotid arteries using ultrasound, and we measured this thing called Intimamedia thickness, which is a particular thickness that you can measure in the arterial wall, which is a good surrogate for atherosclerotic disease burden. And in some patients, you can actually find the presence of plaque as well. This is a hard plaque present in the carotid of a patient. And we look to see, are these blood tests, if we measure MPAs or pro-inflammatory monocytes, is that useful in determining somebody's atherosclerotic burden? So what we've done here is to graph patients as to whether they're low risk, medium risk, or high risk, and we've looked at MPAs and different types of monocytes. And essentially, there's no relationship at all. It's a bit disappointing, really. But having said that, we also found that risk status, according to the charts, didn't actually predict whether they had plaques or not either. So actually, although these charts are very useful on a population level, they don't seem to work that well necessarily when you have an individual patient. But happily, what we did find is that if you looked at patients who did or didn't turn out to have plaques in their carotids, then there was a significant, highly significant difference in the number of pro-inflammatory monocytes in their blood. There were a higher number of pro-inflammatory CD16 positive monocytes in those patients who had plaques. And if you looked at those patients who didn't have plaques, but who had increased Intimamedia thickness, or IMT, there was a very good correlation between IMT and the number of these pro-inflammatory monocytes. So this is suggesting that this particular test, if you can measure CD16 monocytes, might actually be a very good predictor of whether somebody's got atherosclerosis or not. So putting this paradigm into a cartoon here, essentially what we're saying is that monocytes can bind the endothelium directly and transmigrate through and contribute to atherosclerosis. But this whole process can be hugely accelerated if the monocytes bind to activated platelets. Because platelet activation will bind the monocyte, it will transform it into a CD16 positive monocyte, which is more pro-inflammatory, has a much greater ability to bind the endothelium and to cause all the changes that lead to atherosclerosis. So if that being the case, you would expect that if you inhibit your platelets, you should be able to inhibit atherosclerosis. So we went back to a mouse model of atherosclerosis, the ApoE mouse. And what happens is that in this mouse, if you feed it fat over a period of weeks, you gradually get atherosclerotic plaques developing. So this is at baseline, and after 8 weeks, you can measure the number of monocytes in the blood. Now, unfortunately, mice don't have the same kind of monocytes as humans do. But essentially, Ly6C high are the equivalent of the classical type monocytes, and Ly6C low are the equivalent of the CD16 pro-inflammatory monocytes. But you can see that both types increase after 8 weeks of fat feeding, and you can also see that if you also give these animals antiplatelet agents, which are commonly used clinically, either aspirin or clopidogrel, both of those drugs will dramatically reduce the number of those monocytes. Aspirin to a greater degree, actually. If you look at the lesions that develop, actually, clopidogrel only has a marginal response. Aspirin has a much greater response, and that's shown down here. So this is an animal who hasn't been given aspirin, and this is an animal who has, and you can see a dramatic reduction in the amount of lipid that's deposited in those plaques. Is that purely due to aspirin acting as an antiplatelet agent? Well, the answer seems to be no. It's doing something else as well, and this is very recent work where we've shown that aspirin also seems to be stimulating the endothelium to produce a substance called netrin-1. So in the presence of aspirin, you get an increase in endothelial production of netrin-1. And netrin-1 is important because it has a chemorepellent effect on monocytes. Essentially, it keeps monocytes away from the endothelium. And we've done further experiments to show that essentially this happens by aspirin causing acetylation of histones, which are regulatory proteins that surround DNA. And this, essentially what aspirin does, it activates an enzyme called histone acetylase, and that causes histone acetylation. And this is the pathway that we've worked out. Aspirin increases histone acetylation, and that seems to give rise to an increase in netrin-1 expression by the endothelium, which in turn reduces monocyte infiltration of the vascular wall. And we're doing further work to look at the precise mechanisms in more detail. So this is the scheme that we have in mind, that platelets, once they become activated, will bind to monocytes. That binding will cause that monocyte to become more pro-inflammatory and more adhesive to the endothelium. The endothelium will normally produce protective factors which inhibit both platelet activation and monocyte adhesion. And I've talked a lot about nitric oxide, which inhibits platelet activation. There are other substances too, and netrin-1, which inhibits monocyte adhesion. In the presence of cardiovascular risk factors or in inflammation, you get activation of platelets, and you also get inhibition of production of these protective factors. And if you give aspirin, that inhibits, that turns it all around. It inhibits platelet activation, and it increases the production of that protective substance, netrin-1. So that's all good news. Does that mean we should be giving aspirin to everyone? And the answer is no. Because undoubtedly we know that aspirin is effective in secondary prevention. In other words, if you have a patient who's had a thrombotic event already, a myocardial infarction or a stroke, then that kind of patient will undoubtedly benefit from being on aspirin. But the evidence that we have suggests that it's not effective in primary prevention. If you give aspirin to everyone who's clinically healthy, they're not really going to benefit. Because the reason for that is that the benefits are going to be smaller because they're diluted by a whole group of healthy people. And also the benefits are offset by the adverse effects of aspirin. Remember that aspirin is not an innocuous drug. It causes bleeding, it causes peptic ulceration. And in a low-risk population, those adverse effects are going to outweigh the beneficial effects. However, if we could more effectively identify people who are really high risk, who have atherosclerosis, which is silent, those are the kinds of people who would benefit from being on aspirin. One thing I haven't had chance to talk about, haven't had time, is this phenomenon of aspirin resistance. This is where certain people don't respond, their platelets don't respond in the normal way to aspirin. And this is a whole fascinating story in itself. And this is currently being looked at in my group by Dr. Chris Floyd, and I apologize to him, I haven't had time to go through this. It was another fascinating issue that we're looking at. But essentially, what do we do then when we have a patient in our clinic? Well, I've said that the risk charts may or may not be useful. We do them, but I mean, they may not be the whole answer in individual patients. But we reckon that if we do this blood test and measure their MPAs and monocyte phenotype, that actually might give us a very good handle on whether they might or might not have underlying atherosclerosis. And if that blood test was normal, you might be reassured and say no further action is needed. If, on the other hand, it was abnormal, then you might proceed to non-invasive imaging, be it carotid ultrasound or coronary calcium score, CT angiography, and in the future, MRI perhaps. And if that was normal, again, no further action, but if it's abnormal, that's the kind of situation where you might intensively treat somebody with statins to reduce their cholesterol, blood pressure reduction, and aspirin, taking into account whether they might be aspirin resistant or not. So what about the future? Well, if we could modulate nitric oxide, increase nitric oxide over the long term, we would expect that to be beneficial. That certainly seems to be the case in animal models. We need to show that in humans now. And Netrin-1 is a very interesting emerging molecule, and if we can modulate that and increase its levels, again, that might be protective. Aspirin does the job, but as I said, it's not an ideal drug because it does have downsides as well. And if we can develop specific Netrin-1 modulators, that might be a very useful way to go in treating and preventing atherosclerosis. We're also doing some work in our group looking at tracking monocytes using MRI scanning. And again, this is work done by Gabriella Pasquale in conjunction with Professor René Botnar. And they've developed a peptide which is conjugated with gadolinium, which very specifically binds to monocytes. And these are some preliminary experiments. So we've got an ApoE mouse again, which has been scanned at the level of the aortic arch. And you can see that there is uptake of that tracer in the vascular wall there at the site where there is plaque. So these are very preliminary experiments, and we're taking that forward. And if we can do that, that might be a very useful tool, both for research and clinically in the future, for various reasons. First of all, it might give us a good handle on how much plaque inflammation is going on there. If you've got a lot of monocytes going in, you'd expect that plaque to be more inflamed and therefore more at risk of rupture. So it might be a good way of predicting the risk of rupture of a plaque. And also, if we are going to develop novel anti-atherosclerotic therapies, again, it might be quite useful to have this as a surrogate for monocyte infiltration, because, of course, it takes years for atherosclerosis to develop, and you're not going to know whether these drugs work for years and years. But if you can show that they reduce monocyte uptake into the plaque, then that might give you a very good surrogate in the short term to suggest that these drugs are really going to work. I want to finish off now just by thanking my various colleagues. So here we've got Professor Phil Hovienchek and Dr. Andy Webb, my colleagues in clinical pharmacology. These are various characters who work with me in vascular biology at Franklin Wilkins. Professor Jeremy Pearson, Richard Naftalin, Calvin Alfie, Giovanni Mann, Richard Xiao, Sue Brain, Paul Fraser, Luigi Nudi. A very eclectic bunch of characters, but all worth their weight in gold. They're great colleagues, and I've immensely enjoyed working with them, and, very good people to work with. So I thank them all very much. I want to thank the people who've done the work that I've showed you today. I've mentioned Lindsey already, my first research assistant, who's stuck with me for so long. Dr. Peter Okhane, who's now a consultant cardiologist in Bournemouth. Tim Goodman, who I'm very pleased to see in the audience, he did the initial experiment on aspirin resistance. And as I say, I'm so sorry I haven't had time to go through that. Marcy Koch, who worked with Lindsey on the beta-2 work. Nan Chen, who worked on beta-catenin. Ashish Shah and Eugenia Kalia, who worked on the MPA story. Colin Hamid, who worked on beta-catenin and the MPA story as well. Now, these two characters here, this is Professor Biao Xu and Professor Ji Yong. Both of them came to me in the early 2000s on welcome traveling fellowships from China. Biao is now a professor of cardiology in Nanjing University, and Yong is a professor of pharmacology there. And we've continued to collaborate over the years, and much of the work I've shown you today has been a result of close collaborations with these two people. And these are some more senior people who've contributed. This is Val Corrigal, who's helped us with, she's been an absolute gem, helping us with the flow cytometry work. Clive Page, who helped us with the in vivo platelet aggregation studies. Jeremy Ward, who I've collaborated with from the beginning of my time here at King's, initially on the beta-2 receptor work and more latterly on beta-catenin. And Renee Botnar, I've mentioned, who we're collaborating with on imaging those monocytes. This is my current team, Genevieve Vanderslot, Agnesa Mustafa, Christina Warboys, myself, Chris Floyd, Gabriella Pasquale, Ben Ellis. So Dr. Chris Floyd is my PhD student. He's doing the aspirin resistance story. And as I say, I'm so sorry I haven't been able to talk about that today. And he's being supported very ably by Agnesa, who is an undergraduate student. Christina, my postdoc, funded by the BHF, who's taking on the beta-catenin work and being aided and abetted by Genevieve here. Gabriella was my PhD student, now our clinical lecturer, who did the monocyte phenotype work, and more lately she's working on the monocyte tracking experiments as well, and being assisted by Ben Ellis here. The other guy you see here is not a member of our team. In case you don't recognize him, he's John Keats, who you know better as a romantic poet. What you may not know is he actually trained at Guy's as a surgeon apothecary, but found it all too gruesome and terrible, so he changed careers and became a romantic poet, which I think was probably the right thing to do. I'd like to thank our funding bodies. We've had a fair amount of funding from the British Heart Foundation. We're very grateful to them, and also to the Guy's and St Thomas's Charity, who've been very generous to us. And we've had project grants over the years from Diabetes UK, the MRC, and the Wellcome Trust. I want to say a word or two about my family, who are very important to me and, the bedrock of my life, really. I often found it difficult to say the words that I feel about them, so I'm going to quote somebody else's, Winston Churchill, who said, "There is no doubt that it is around the family and the home that all the greatest virtues, the most dominating virtues of human society, are created, strengthened, and maintained." Well, you can't say further than that, and I think, it sums it all very well for me. I want to thank my mum and dad, who are here at the front here. I just want to say to Mum and Dad that you've been an absolute source of strength to me, and I can't thank you enough for everything you've done over the years, so thank you very much. This is my brother Jack, who is not only my brother, but my friend, and he's also here, and it's lovely to see you, Jack. This is my wife, Alethea, who, as I say, I met through Graham Jackson. She's been an absolute joy to me, a source of strength, a support all the way through my career. And I really can't say how much she means to me. You saw these characters before, Joshua and Louis. They've grown up a bit since the previous picture. This is Joshua, this is Louis, both looking very cheeky, and they're approaching their 5th birthday, and I'm glad they've managed to make it today, especially since tomorrow's a school day. So great to see you guys. I've got more involved with Josh and Louis' school. One of my other passions is astronomy, and I'm a bit of an amateur astronomer. So I offered my services to the school. I could say I could talk about medicine, or said, well, no, I hear you're an astronomer. Talk to us about that. They're obviously more interested in that than in medicine. So this is me talking to the reception kids about astronomy. It looks like I'm talking about Saturn there, and I think that's the Earth there, being very attentive. I belong to this group of amateur astronomers called the Baker Street Irregulars. And we meet once a month in Regent's Park, and this is them. I've learned a hell of a lot of astronomy from them, so I thank them very much. I think one or two of them are actually going to come tonight. Can't actually make out whether they're here or not, but thank you, guys. And only last week I gave another talk on astronomy to the Year 1 kids at their school. This is a lovely card they sent to me, which means a lot to me. It's going to go into my appraisal folder for next year. It's very different talking to young kids to talking to medical students and science students. It's both very interesting and very challenging, but very different. And I found both of those very, very rewarding indeed. Anyway, I'm going to stop now, and thank you all very much for your attention.
I now call on Professor Hovienchek, Professor of Clinical Pharmacology, to give the vote of thanks.
Well, thank you, Albert, for a truly wonderful lecture. I think that your elegant studies on the platelet monocyte interactions have given us really new insights into processes of atherosclerosis, and I'm sure are going to lead to new approaches to treatment. I think the other thing that came across in your lecture, and what's certainly apparent to your close colleagues, is that your leadership and inspiration to younger researchers, many of whom are with us this evening, so that's absolutely wonderful. And in fact, Albert's leadership extends outside the research laboratory. So Professor Greenhoff has already told you that he runs not just one, but two major courses, BSc in clinical pharmacology and translational medicine, MSc in clinical pharmacology. And Albert and colleagues have been responsible for getting some major European funding and for King's being recognised by the European Centre for Innovative Medicines programme as a European Centre for Excellence. So to sum up, I think that we've heard great science, and Albert is a great leader, and this is just an inaugural lecture, so there is so much more to come. And I think that Professor Ferro, his star is in the ascendancy, and with his team, I hope we'll be seeing, and I'm sure we'll be seeing, many future academic grand slams. Thank you, Albert.
Ladies and gentlemen, I'm sure you'll agree with me, we've heard a superb inaugural lecture. Please join us celebrating the many successes of Professor Ferro's career in a drinks reception in the foyer. I look forward to meeting you all there.